So for our last speaker, I am extremely excited to introduce Professor Andrew Ward. So Andrew is a structural biologist interested in vaccine design and is a professor at Scripps Research in California, USA. He leads a group of scientists that use cryo-EM and computational approaches to unlock the molecular details underlying antibody-mediated neutralization of viruses and pathogens. And these atomic level observations have enabled the design of improved vaccine candidates against HIV, influenza, coronavirus, Ebola, malaria, and other pathogens.
Thanks for the invitation. It's an exciting time to speak with you and share some of the work that we did in my lab in collaboration with other investigators that really led to the development of some of the COVID vaccines that are being deployed today. So my lab is in La Jolla, California, so just north of the Mexican border. And we're at the Scripps Research Institute, located right next to the Torrey Pines Golf Course. And this is where my lab is. And inside this concrete bunker of a building is our cryo-EM suite, where we do all of our structural biology. This is really the centerpiece of the whole facility. That's managed by Bill and Hannah, and it's been about a $25 million investment over the past 10 years or so to really make this into a high throughput molecular atomic pipeline to solve structures involved in everything from neurodegeneration to vaccines, which I'll highlight today. Behind the scenes, we've also done a lot and really employed the best and newest in high performance computing. And this is an effort managed by JC and Charlie. And as Charlie likes to say to people, you know, if you were to print out all of the images we take on these microscopes, it would be about 1.25 feet of pictures per second, which would add up to Mount Everest in about 6 hours. But we pack that all into these, 7-inch hard disks. So we generate terabytes of data using this cryo-EM facility, all to solve structures, for my lab's case, of pathogens such as HIV, Ebola, malaria, influenza, and coronavirus. And in most cases, we're looking at how antibodies bind to and neutralize these viruses. From there, we can learn lessons about how to design better vaccines and really focus immune responses to the critical pieces. So this is really in contrast to historical vaccines and many of the childhood vaccines that you may have received. So polio vaccine, even though it's very highly effective, is a pretty simple solution. And that is to grow up a bunch of polio and then kill it, inactivate it with some pretty nasty chemicals like formaldehyde. But when this is injected as a pretty dirty mixture, you develop lifelong and durable immunity. For a lot of pathogens such as HIV, influenza, coronaviruses even, we can't just heat up the virus and kill it. For flu vaccines, we do that, but it's the efficacy isn't quite great, and there's a lot of problems. Some of the coronavirus vaccines have also taken that approach, but really the best vaccines are the ones that just take the critical piece of the virus, stabilize it, engineer it, and using the structures that we and others generate to create these vaccines that can be effective really on a global scale. So this is known as rational vaccine or vaccine design or structure-based vaccine design. So for coronavirus, which is obviously the topic of the moment, the key protein is the surface spike protein, this red protein. And this red protein is responsible for recognizing host cells and then fusing into host membranes, as I'll show in a video coming up. And Haley in my lab has done she's a graduate student, and has done a really great job of drawing pictures and making illustrations in Adobe Illustrator to come up with this Science Simplified series on our YouTube channel that really disseminates a lot of, or distills a lot of complex knowledge into an accessible platform. So I encourage you to check that out if you're interested. So this is actually the protein, the spike protein, and really the gymnastics that it undergoes in order to fuse. So here's the spike protein. This is known as the pre-fusion conformation. There's the S1 domain and the S2 domain. So the virus is floating around. When it finds a cell that expresses this ACE2 receptor, it binds, and then you can appreciate these large-scale conformational changes that happen. And the purpose of these conformational changes is to bring the viral membrane close to the host membrane and then eventually fuse them so that the RNA genome can get in the cell and the virus can replicate. So the role of a vaccine and antibodies is to really intersect and intervene and prevent that process from happening. So we started working on this in about 2015, and a good friend and collaborator, Jason Mcclellan, had contacted me. He knew I was using cryo-EM, which was really like an up-and-coming technique at the time, and he had an interest in coronaviruses, and we both had an interest in vaccine design. And it's one of those chance interactions. He sent me an email, and I said, "Sure, let's go for it." And I said, I hope we're hugely successful and publish lots of high-impact papers, so on and so forth. And of course, we didn't appreciate at the time, the impact that the research would have. But this is kind of, one of those cool anecdotes in science and how things proceed. So we were interested in coronaviruses because we knew that there were pandemic potential coronaviruses that with high lethality, like SARS and MERS, that were coming up about every 10 years. And then there are a group of seasonal coronaviruses, which you've probably never heard of until recently, that cause cold and flu-like symptoms. They're not really problematic unless you're immunocompromised or elderly. So even though we were very interested in the pandemic ones, they turned out to be very difficult to characterize. So that spike protein, which we knew was going to be the focal point for vaccine design, really couldn't be made and analyzed using our structural biology techniques. So we ended up looking at HKU1, which was a pretty distant relative. Here's sort of the family tree of coronaviruses, and most of the ones we care about are in the beta genus, and then within that, HKU1 is this in the A family. So through a lot of biochemistry and hard work by Jason's lab and my lab, in collaboration with Barney Graham, we're able to use cryo-EM to solve this structure of the coronavirus spike protein trimer. And here's what it looks like. So now we have an atomic level detailed picture of where all the domains are and where the fusion machinery is. So this is where it binds the receptor, and this is that machinery that undergoes those large conformational changes to fuse the membranes. And so we could use that information to start designing and changing the protein, mutating different regions to really stabilize that pre-fusion and locked-in conformation. And here's just showing you, after screening, 50 or 100 different mutations and using this sort of rational approach, something that really didn't express, that couldn't be made before, so the SARS and MERS wild type, all of a sudden could be made at high yield and high quantity. And then when we looked at it under the electron microscope, it went from a mixture of these pre-fusion globular to these post-fusion extension conformations. And then we add those 2P mutations, it made the sample very homogeneous. And so we knew we were onto something. And it turned out that these mutations placed in this particular region would have a positive impact on any of the beta coronavirus spikes that we looked at. And so, this got published and was on the shelf really and ready to go since 2017. And then when the pandemic hit, we had laid out the blueprints through publications, and companies who also appreciated that the spike was going to be the key antigen for delivery, started incorporating these mutations into their vaccine platforms. And here I'm just showing you, this is from Novavax. Novavax is one of the next vaccines that will get approved, and we showed structurally that it was beautiful and well behaved. So this is this nice arc of how basic research really informed vaccine design. And then these mutations have been incorporated into the vaccines for Moderna, Pfizer, BioNTech, Johnson & Johnson, Novavax, CureVac, and others. And it really shows you how robust these mutations are in terms of stabilizing the correct form of the antigen. So the design, the typical workflow that we undergo is solve structures and do computational design. There's a number of ways now to deliver, and luckily, most of these platforms were available. So really all that you had to do was plug in the right sequence of the spike protein into any one of these different vaccine platforms. And then the typical workflow is you do a lot of iteration where you analyze neutralization and structure and antibody sequences. But fortunately, we only had, basically the companies only went through this loop once, and were able to rapidly down select. And then the focus really became on this phase of manufacturing and at scale. And so this is not challenging, right? It's easy to do a lot of this at small scale in the lab, but to make hundreds of millions and billions of doses is a challenge. And certainly, in the first world, that problem has been, largely addressed. But in the third world, in harder to reach communities, this is still a major unmet need, and it's really here. So we got fortunate that the science we and others did really informed this process and allowed a vaccine to be generated in less than a year, which I think nobody predicted would happen. And not only one vaccine, but many vaccines. And so now the effort is really to look at the antibody responses to these vaccines, where they bind on the spike protein, how they neutralize, to make even more efficacious vaccines, and really address the variants of concern and other things. So sorry on the tight time and the quick run through here, but just to sort of reiterate, the prior knowledge and the risks and chances we took to look at coronaviruses more broadly, and as I mentioned, a seasonal coronavirus, really provided the details in terms of structure, stabilizing mutations, to intersect with vaccine platforms that had been being developed for different reasons, and mRNAs actually were being developed for cancer therapies. So these things all were combined. This prior knowledge was combined in really rapid order to generate what are remarkably effective vaccines. And of course, the genome coming out of China very early was critical to know the sequence of the spike and other proteins. So again, this ability to do fast science with a lot of high-end technology and capitalizing on prior knowledge is how we were able to address this pandemic in shorter order than I think anyone predicted. And I think, optimistically, the lessons learned now will be used to develop vaccines against variants of concern, maybe a pan-coronavirus vaccine, future emerging pathogens, or even improve seasonal influenza or other vaccines. So just to end and acknowledge, this is a really fantastic collaboration with the labs of Jason and Barney. This is the original coronavirus structure team from my lab that did the original work. And then we have a team still working on coronavirus research. We've had generous support from National Institute of Health, the Gates Foundation, and Scripps Research, which really, fund these high risk, high reward type endeavors in my lab. And then just to end and say, here's the full picture of the lab. It's a really fantastic and diverse group of scientists doing lots of different things. And just to also note and intersect with this conference a little bit, we've had a great set of interns from 16 years old to about 22 years old, high school and undergraduates. And Scripps has done a really nice job of bringing these scientists in the lab. And the really fantastic part about it is, we try to inspire them, and many have gone into science careers or medical careers. And two, Reed and Alessandra, have actually started in high school and now are in college and have come back to the lab. So it's really great to engage young scientists, inspire them, and then see what they can do in the next phase of their career. So with that, I'll end, and thanks for your attention. Sorry for the difficulties getting this started.
Great, thank you so much, Andrew, for that talk, and it's really inspiring to hear how your research has sort of driven and influenced vaccine design presently within the pandemic. Not sure if we have enough time for questions, but there is one I can ask now. So Karina asks, how did you get into cryo-EM in the first place and sort of the structural biology?
So to answer that question, it's a really nice story. When I was an undergraduate researcher at Duke University, just working in a lab, I was working in an electron microscopy lab looking at insect cell muscle architecture, and there really was no cryo-EM at the time. And it worked for a team, a husband and wife team, and I didn't really know what I wanted to do at the end of college. And they said, we have a colleague at Scripps in San Diego, and he's trying to do something called cryo-EM, and we think this is going to be really cool, and it's probably going to be a big deal. And they basically called up Ron Milligan and said, "Can you hire this guy?" And so I packed up my bags 2 weeks after I graduated, headed across the country, and ended up in what happened to be, really the early stages of cryo-EM. And just fell in love with EM and structural biology in general, and it kind of just followed my nose from there. So it was a little bit of luck and a little bit of timing and just being in a great environment.
Great, thank you so much for that answer. There are a couple other questions in the Q&A, if you don't mind just typing out some responses to them. But thank you so much for joining us, and it was really great to hear about your work.